Your objectives for this section are to compare and contrast the two rounds of division in meiosis. Remember, mitosis only had one round of division, but in meiosis, you're supposed to end up with 4 haploid cells, so you got to divide twice to do that. And then we'll talk about how haploid cells develop into mature gametes. Right, the whole process of meiosis is to make gametes, sex cells. So obviously, those go hand in hand. We'll get started with this. And in this first slide, homologous chromosomes versus sister chromatids. I just want you to draw the picture that I'm drawing because I want to make sure that it's very clear the difference between what is a homologous chromosome and what is a sister chromatid, because they mean very different things. This is one chromosome right now, but it has 2 chromatids. So remember, this side and this side are identical copies to each other. Those were the sister chromatids that we talked about before. And those are identical to each other because they're exact copies of one another. They were made as a copy during that S part of the cell cycle. But when I say homologous chromosomes, I mean 2 different chromosomes So you're seeing 2 different colors here, obviously. But those 2 chromosomes should contain the same genes. So if this chromosome, the blue one, and the red one, they both have that gene that codes for eye color. Right? You can have blue eyes, maybe that's the allele that's going to be on this chromosome, but maybe the brown-eyed allele is going to be on the other one. You got one of those chromosomes from mom, one of them from dad, but each of them code for the same thing. You're not necessarily going to see blue eyes if you have a brown-eyed allele because that brown eyes is dominant, like we'll talk about later, but you have 2 of everything. 2 chromosomes. These homologous chromosomes have the same genes on them, although they will not be identical. They likely have different versions of that same gene. So one might say blue eyes, the other one says brown eyes. This one might say that you're going to have curly hair. This one says you have straight hair. Because ultimately, all of those different traits come down to proteins. And those proteins come from your DNA. So they go together because they have the same genes, but that does not mean that they're identical. They are actually different. But they have, this one might have the blue-eyed allele again, this one has brown eyes, but they both have eye color, that gene on them. That's why they're homologous. That's why they go together. So when I'm saying homologous pairs, that's talking this and this are going to be homologous as long as they have the same genes on them. So they'll have the same genes. I should probably switch to black. That's going to be hard to read. But they are not necessarily identical. Now that being said, you might have 2 blue eye alleles. Right? If anyone in this room has blue eyes, you do have 2 blue eye alleles. One from mom, one from dad. So they could be identical, those genes, but they don't have to be. There could be a different version on one versus the other.
Isn't brown eyes dominant?
Yes, it is.
Is that like 4 times?
But if I look at this side versus this side, when I'm looking at the chromatids in this chromosome, if I have the blue eye allele right here on this chromatid, I have the exact same thing on this one because those are identical, for sure. But I don't have to have the blue eye allele on the red chromosome, although it could be the blue eye allele. It just depends. What did you get from mom or dad? So homologous chromosomes have the same genes, but they're not necessarily identical, whereas the sister chromatids, one side of a chromosome versus the other, those are identical. Those will be exactly the same, generally speaking. So that's just trying to clarify that, make sure that we understand it before we go, because that's a big point. That's something that you need to understand. Okay, so meiosis includes 2 rounds of division. In the first round, homologous chromosomes are going to be separated, and you're going to end up with haploid cells. So the key to that first round is I'm going to cut that chromosome number in half. So I might write that in here. In the second round of meiosis, sister chromatids of replicated chromosomes are going to be separated. So actually, this second round is pretty much the same as mitosis before. If you're separating chromatids, that's the same as mitotic division. Whereas when you're separating the homologous chromosomes, that's meiosis specific. Right, so we'll have a couple different pictures to follow with later. But in that first round of division, there's a huge difference when you're separating the homologous chromosomes versus the sister chromatids. So that takes us to meiosis 1 here, so we can see all the information. Again, this is not necessarily that useful. We probably want the picture instead, so we'll draw a picture to go with this. But if you look at number 1 here, prophase 1, the nuclear membrane breaks down. That's what happened in prophase last chapter. Duplicated chromosomes begin to condense. Chromatin turns into chromatids. That happened last chapter too. The only thing that's new is the only thing I'm going to highlight. Those homologous chromosomes begin to pair up. So that did not happen in mitosis. It is happening here. So again, we're going to draw some pictures with this. And I got a little bit of a head start on you, and I understand you're going to have to take some time to catch up, but that's okay. I am going to do all the way prophase, metaphase, anaphase, telophase for this first round. I have to do another prophase, metaphase, anaphase, telophase for the second round. So make sure you have lots of room somewhere to work with. But this first cell here, in this prophase, the only thing that's different from before is I'm trying to show you that there's homologous pairs that are matched up there. Right? This blue goes with red, just like they did earlier in the notes. So if you don't have different colors, that's okay. But I did try to make the blue and red a little bit bigger than the orange and the light blue. So they're just larger chromosomes so that you can see obviously the little ones go together, the bigger ones go together. But that's the only difference in prophase 1, and it makes a big difference in metaphase 1. So again, if you don't get this all at once, it's okay. We can get caught up, plus this is recorded for later. So the first difference, homologous chromosomes begin to pair up. Metaphase 1, going to look different than it did before because I have paired homologous chromosomes that are going to line up in the middle of the cell. So instead of having all of the individual chromosomes lined up in the middle here, I have pairs lined up. Right? Red with blue. Because red and blue hold the same genes. Not necessarily the same version of that gene, but they hold the same genes. And the orange and the light blue go together. So you still see things lined up right in the middle of the cell, but they're pairs lined up in the middle. Still have centrioles, spindle fibers, whatever else, so that you can pull those things one way or another. But that should look very different than mitosis before. Mitosis before, I'd have the red, blue, orange, light blues all stacked up, just one right on top of the other. But now I have pairs that are stacked up in the middle. Because I'm going to take this red one, move it to this side. The blue one needs to go to the other side. They need to be separated in this first round of division. Which brings us to our next point here. That alignment in metaphase is completely random. The way it's drawn right now, blue's going to go with orange and red's going to go with that light blue color. But that doesn't mean I had to have it that way. I could have drawn it, or I could have aligned this so that the orange was on this side and the light blue was on that side, or I could have flipped the blue and the red up on top, and it doesn't matter. That's completely random. But you understand, because that is random, it will give you different options at the end of the day. You could have the blue go with the orange. You could have the blue go with the light blue. You could have red go with blue, light blue, or red go with orange. There are 4 different combinations that could result from this because there are 4 chromosomes. You can actually do the math for that. Remember, this organism to begin with has how many chromosomes, Corbin? How many chromosomes do you see? So 2n equals 4. I know. So then n equals what? And to do the math to figure out how many combinations there are, this one's easy enough to do in your head. I know that the blue can go with light blue or go with orange. The red can go with light blue or with orange. That's 4 different combinations. But to do it mathematically, you're supposed to do 2 raised to the nth power, which means in this case it's going to be 2 squared, which is 4. But if it's not a simple organism like this, if it has more chromosomes, say 2n equals 46, because we're talking about a human, what's half of 46? Does anybody know what 2 to the 23rd power is off the top of their head?
Yep, I do.
Boy, Jake, what is it?
Well, it is 8,388,608.
And he's correct. He's the smartest calculator in the room. Or else he just noticed that I put that number right here. So 2 to the 23rd is this huge number here. So for a human doing meiosis, there are over 8 million different combinations, which means if you actually had to draw this, it would take some time to come up with all those different combinations, right? So I'm not going to expect you to do that. That doesn't make any sense. But understand that although we make it look really simple in a cell like this that only has 4 chromosomes, when you have more chromosomes, there's more possible combinations. And also understand what that means. I'm going to highlight this a different color for a reason, so if you can, do it. I want it emphasized. That random alignment is why sexually reproducing populations end up with such a huge diversity of genes, right? There's always this random shuffling, even when it's just one person making those gametes, there are millions of combinations. This is why even offspring of the same 2 parents are always going to be very different from each other. They can't be exactly the same, unless they do happen to be identical twins. We already talked about that, right? But identical twins, they started out as the same cell, and then became 2 different cells by accident, pretty much. But to have a sperm and egg fertilize an egg separately from another sperm and egg that are fertilized and have those be exactly the same, that is, statistically speaking, improbable, which is a mathematician's way of saying impossible. Right? You can't have 2 yous. Your parents would have to have over 8 million kids to have another one of you the exact same as you, right? And that being said, this is only with 1 parent. What's 8 times 8? And 8 million times 8 million is 64 trillion. Your parents would have to have 64 trillion offspring before they could have another one exactly like you, which I'm not sure if you understand how that works with humans, but that's not likely. Even spiders don't have that many offspring in their lifetime, and they have thousands at a time. Yes?
So if you had like 2 identical twins, and then another family with identical twins, and then they had kids, would they have this identical kids?
They could.
They could, but they don't necessarily have to
They don't
because
have
of this. That is a very good question because that emphasizes it though. So you have two identical boys, two identical girls. They get married in those pairs, have kids. You still would never have identical offspring as long as it's normal meiosis. If the same error happened again that did before, you might have identical twins again. But those two different couples could never have this. They'd have to have literally that 64 trillion different kids. That was a good way to put it, actually. It's confusing, but so it can't be an identical twin in that case. There's no way it could be. But there were the right genes available to make an identical twin. I gotcha. I like that. I'm gonna actually use that later because that emphasizes the point pretty well. Anybody else follow that? Do you understand what we're talking about? Because that was a little weird. I'll try to explain it later if you need some help. Okay, so random alignment was the big deal. Otherwise, anaphase is the same as it was before. Or it looks similar. Right, those spindle fibers are going to shorten, so you have those chromosomes moving away from each other. You should actually be able to tell that they're moving because they're kind of folded over as they get pulled through that liquid. Otherwise, in the specific combination that I put up here, red is going with light blue and blue is going with orange, but that doesn't mean that's the way it had to happen. It could have been red with orange and blue with light blue. And then finally, this last picture is a telophase / cytokinesis. So I'm going to put those chromosomes where they belong inside of a nucleus now. So I got a red one, and I got that light blue one. It's a little bit smaller than the red one. I have an orange one over here. It's a little bit smaller than the dark blue one. But here's the next major point of emphasis. Corbin already told me that this one has 4 chromosomes. Two N equals 4. When I look at this new cell that's being formed right here, Mary, how many chromosomes does that have? Just the 2. So I'm not at 2N anymore. Now it's N. That's when my chromosome number gets cut in half. Since half of the chromosomes went this way, half of them went that way, I'm going to have 2 new cells with half as many chromosomes as the original. That's when I go from being diploid to haploid. Even here, technically, I have 4 chromosomes in the same cell. But after this cytokinesis over here is complete, right, and those are 2 separate cells, there's only 2 chromosomes in each nucleus. So I went from 2N to N. This would still be 2N. This would still be 2N. After that cytokinesis is over, I'm now at that N chromosome number, which was the whole point of meiosis anyway, was to get from 2N to N. But this is only the first round of division. I got one more to go. So in this, I need some more labels. This is metaphase 1, anaphase 1, telophase 1 / cytokinesis 1. For my next picture, I'm just going to use one of these 2 cells, but you got to understand that both cells would divide again. I just don't want to draw both of them because that's just going to take twice as long. I don't really want to do that. The last thing to highlight on this last slide, though, before we go, is that emphasis at the end there is that you did end up with 2 haploid cells instead of 2 diploid cells, like you would have in mitosis. So that chromosome number got cut in half. Otherwise, everything else, nothing new. Not really. Okay, so this next round of division. Meiosis 2. I'm just going to start with the red and light blue. It really wouldn't matter which one I did. But I have that cell that needs to divide ultimately. So in here I got red and a light blue. The light blue is still a little bit smaller than the red, so I can tell the difference. This looks like a normal prophase. Otherwise, needs spindle fibers, that kind of stuff. Notice it does have a Roman numeral 2 next to it because it's not the first prophase, it's the second prophase. You're going to know the difference. But from here, all these 2s in the meiosis 2, those are pretty much exactly the same as mitosis. This next step is going to look the same as it did before. Right up and down the middle of the cell, I'm going to have my chromosomes. I only have 2 chromosomes now, but they're just lined up. They're not paired. They're just stacked up right up and down the middle like they were supposed to be from last chapter. Still going to have centromeres connected. And I'm going to divide those sister chromatids now. Right? 1/2 of the X is going to go one way, 1/2 is going to go the other way. So anaphase over here. Got half of the red going this way. The other chromatids going the other way. Same with the light blue. And so in my last cell, that telophase cytokinesis combination, I'm going to have one of each chromatid, one red, one light blue. Red's still a little bit longer than light blue, so I can still kind of tell the difference even if I'm not coloring. On each side. Probably need some labels. Remember, this is metaphase 2, Roman numeral 2, anaphase 2, telophase 2 / cytokinesis 2. But at the end of the day, when you look at this picture, it looks the same as the picture last chapter, right? But there is technically a difference. Because last chapter in mitosis, you started with diploid, you ended with diploid. Was this diploid to begin with? No, it was haploid. I had that n number of chromosomes here. And I ended up with 2 haploid cells at the end of that. So the chromosome number didn't change. That's why it's similar to mitosis. But it's not really mitosis because I don't have 2n and 2 things with 2n at the end. So it's similar, but it's not exactly the same. Right? That's the only real difference that you can see, that there's not as many chromosomes as there should have been. Now remember, that happened to this cell, but it also happened to this cell, so you have 4 haploid cells total at the end. But that's basically the whole process. So hopefully you got your pictures in. If not, again, you can look at this online later, just kind of fast forward to the spot where you need to draw, or there's a picture in the book that goes with that pretty well too if you wanted to look at it later. But this is meiosis 1. The whole purpose of meiosis 1 was to cut that chromosome number in half, go from 2n to n. This is meiosis 2. I started and ended as n as well, haploid from the start to finish. But I did separate those chromatids. I have to do that still to finish this off. That'll be good. So this was the meiosis 2 description. The only thing you need to highlight maybe, I ended up with 4 haploid cells because I started with 2 different cells that were both going to do this, right? 2 different cells went through this meiosis 2 to give me 4 cells total. but they were also haploid to begin with, so they ended up being haploid. Otherwise, some key differences up here. Meiosis has 2 cell divisions. Mitosis only has 1. Right, so fairly simple blanks to fill in. Meiosis here results in haploid cells. How many haploid cells? 4. So you might put that number in there just so you can clarify. Whereas mitosis is going to end up with diploid cells. How many of those? 2. So if you put a number with that, that might help a little bit. Otherwise, due to our shortened class periods today, I think that's kind of where we stop with the notes for today.